Hey guys, and welcome back. So today we're going to be talking about how to get a perfect 10 out of 10 on your biology data test, which is coming up for a lot of you. I have already made how to get a 10 out of 10 on the physics data test video, so that will be linked below, and chemistry will be coming sometime next week. I do understand that data tests are coming up super quickly, so we need to get in as much practice time as possible. Now, if you do want me to make any additional videos, like maybe there's a specific question type that you want me to go over or anything like that, please let me know in the comments and I will make a video for that one. The other thing that I want to touch on is there are a gazillion resources in the QCE Discord server. That is where I got a lot of these screenshots I'm using today for, and if you ask me to do specific questions, that's where I'll get my resources from. It is super duper important that you join up in that server because there are literally so many free resources in there. And you can literally talk to anyone in that server to ask for help anytime. Everyone's pretty nice. I'm also in there, so if you do want to ask me any personal questions, if you head on into the server, you'll see me floating around somewhere. I'm under the same username, Imogen K, so you should be able to DM me. Other than that, I'm just going to pop the recording on and then we are going to get started. So the first thing that I really do want to touch on when it comes to biology is the whole formula thing. So in physics and chemistry, you do get like a whole formula and data booklet, but in biology, you literally do not get anything like that. Now, there are not a whole lot of formulas that you actually need to use in biology. There are only two that are actually stated in the syllabus to the best of my memory, and they will be relevant for this data test. And then there are a couple more that are also relevant for the data test and not really anything else except for your IA2. But yeah, so all the formulas in biology are definitely only for ecology. They aren't given to you, but there aren't too many that you need to know. So we are going to go through a couple of them today, along with some other stuff. So starting off with the formulas, the formula for species evenness is as follows. So to calculate your species evenness, you want to have the number of organisms in that species and divide it by the number of organisms in total in the ecosystem, and then you will get some sort of proportion there. Now that is exactly what you want because species evenness, which can sometimes also be called relative species abundance, is actually a measure of the proportion of each species in the area. So that means for each species, you will need to calculate a proportion using this formula every time for each species in the area. Now, obviously in your data test, they're not going to be very realistic and they're not going to have literally hundreds of species in there. It might only be like 5 species, so you might only have to quickly calculate it 5 times or something like that. Super, not hard at all. You just have your calculator and you can calculate that all. But I will warn you that because these are proportions and the number of organisms in the ecosystem is the same every time, so nothing's changing there, you should have all of your proportions, so all of your calculated species evenness, if you've done it for every single species in the ecosystem, it should add up to 1. So if you have done it for all species in an ecosystem and it is not adding up to 1, it's more, it's less or something like that, you have miscounted somewhere and you've done something wrong. So that is just a nice little check that you can use if you're a little bit concerned about whether or not you've done it correctly. Now, a more common or a more likely formula question that you will get in here, sorry, I meant to say that species evenness, I doubt they're going to make you calculate it. Typically, you would be given it and you need to make statements based on that. But this one here is the Lincoln Index, and this one is all about estimating a population size. So if you were given this data about a population or whatever, this is what you use to estimate a population size. So what happens is some scientists will go out and they will catch a whole bunch of organisms or organisms of a species, and then they'll find some sort of way to mark them or tag them. They'll then release them back into the wild, give it a couple of days or however long they think is appropriate, and then they catch them all over again and see how many in the second lot that they caught are marked. And then we have a special formula here that can help us figure out a population estimate based on that. So here we've got our formula. Now big N, so capital N for November, is our estimated population size. So that's what we're trying to figure out. Capital M for Mike is the number of individuals captured and marked. So when that scientist initially goes out into the wild and catches like a bunch of animals, maybe he catches 10. Let's just say he catches 10 of these animals and he puts a mark on all of them or tags them in some way. He then leaves it a couple of days or however long and he goes back out there. Lowercase n for November is the number of individuals captured the second time. So he goes back out there and he captures, this time he captures 8 animals. Then he has a look at these 8 organisms that he captured and he checks how many have a tag on them. So in this case, lowercase m, so lowercase Mike is the second, out of the second bunch of things he caught, how many had a tag on them. So he's caught these 8 ones the second time and he has a look and actually only 2 of them are tagged. So then we put that all together and we would get 80 divided by 2 and then we would get 40, which means our estimated population size for this specific organism is 40. So that's the whole idea behind that one. They probably will actually give you the formula for this one and the SDI because they are a little bit more complicated. From what I've seen in a lot of the official resources and just other resources is that they actually give you the formulas and they state what the variables mean. So I really do recommend that you do remember them by heart, but just in case something goes wrong in the exam, you panic, something like that, a lot of the time it actually is there stated for you. Now, I do recommend showing your working out, like to the best of your ability, just in case something goes wrong, because then you can snag a follow-through error. So that might mean going, oh, okay, so I'm going to do m equals 10, n equals 8, and then little m equals 2, and that just shows that you've written down the variables, so you did identify the correct numbers for what variable, but maybe you just made a calculation error and somehow said that there was actually 60 in the population, and that's just a calculation error. So then there you should get some sort of follow-through error because you did state that you knew what each of the variables were, and maybe you substituted it into the formula correctly, you just somehow went wrong in your calculator and didn't have time to check. So I really recommend making sure that you write everything down. The other thing that I would highly doubt that they would ask you when it comes to the Lincoln Index, but I will just let you know just in case, is in the case that they ask you what the requirements or like rules are about doing this population estimation, something that I would definitely be writing down is, first of all, you need to make sure that you don't wait long enough that reasonably organisms in this species could die or reproduce and migrate. So we want to make sure this population is as contained as possible. So you need to give the organisms enough time to mix back into their population, but also you need to make sure that there aren't a significant amount of organisms leaving, dying, or being born, just to make sure that we aren't changing the population too much. So usually that is about a week or whatever, just depending on the organism, just to make sure that you aren't skewing your data in any way. I doubt they would ask you that, but since this is a data test, there's like a slim possibility that they might ask you about the processes behind collecting this data. Another really important formula is Simpson's diversity index, and this is probably the most important formula in all of year 12 biology. Now, what is Simpson's diversity index and what does it actually mean? So I'll start off with this formula, and I'm just going to cross out that little sigma down there because it causes confusion. So we've got this SDI, and this SDI formula is 1 subtract this whole big fraction here. So let's break down the fraction bit by bit. So this sigma here, sigma means sum of. So that means we need to be adding lots of things together. So what we're doing is we're adding together lots of these n minus 1 times n. So lots of these. And what does n actually mean? So n is the number of each species. So say we have 5 different species, you need to add together 5 of these different n bracket n minus 1s all together, where n is the number of species in each of those 5 species that we've got here. So n is the number of organisms, I should have said, in each of these 5 species. So you calculate that n bracket n minus 1 for all of them, and then you add them all together. Then you would divide by this little number here. So big N is the total number of organisms in this ecosystem. So we would do big N, say let's make big N 30, and then you do 30 minus 1, so that would come to 30 times 29, and that would be on your denominator there. Now, you will get an answer somewhere between 0 and 1, where, just like a probability, because this actually is a probability, which I will get to. But Basically, that means the higher the SDI, so the closer to 1, the greater the diversity of this ecosystem, and then the lower or the closer to 0, then the diversity or biodiversity isn't actually that good. Now, what does Simpson's diversity index actually mean? And that is super duper important. Simpson's diversity index is the probability that 2 randomly selected organisms from a sample will belong to a different species. So if the probability or the SDI is higher, that means that there is a greater probability of you selecting 2 random organisms that they belong to different species. So that means it does have a greater biodiversity. Now, there is something else we need to consider that might pop up, and that is actually Simpson's index. So Simpson's index is the same as Simpson's diversity index. We just remove the 1 at the beginning. So instead of doing 1 minus this massive fraction, instead we've just got this massive fraction. So this is going to show us the opposite, if you understand your probability, the opposite of what Simpson's diversity index shows. So Simpson's index will instead show the probability of 2 individuals that are randomly selected from a sample being from the same species. Now, I doubt you'll have to do much with Simpson's index, but just make sure you know the difference between Simpson's diversity index and Simpson's index and how they are inverses of each other. Now, I really recommend practicing a lot of these SDI questions because they actually are pretty lengthy. Like, there is a lot of little tedious bits of maths involved, and lots of mistakes can be made in these. So I really recommend in your data test spending the time on actually stepping out every single calculation. So do your n-1 the 5 times and write that out 5 times, and then write out you adding it all together. You need to be really, really careful that you don't miss anything here, and you need to show you're working out to prove to them that you actually do know what you're doing. Now, there is a major question type that comes on with these SDI questions that sometimes people get a little bit frazzled by if they don't know what SDI actually means. Some people might just go, oh, well, SDI shows the biodiversity, so the greater it is, the better the biodiversity. But if you don't know this definition about randomly selecting individuals and then belonging to different species, then you're going to have a hard time answering a really specific kind of question, which is this. Explain the probability of randomly selecting 2 individuals that are different species from the terrestrial ecosystem. A lot of people are going to see that and go, how do I know the probability of randomly selecting 2 different species? Like, what the hell? How do I know that? SDI, it is literally just your SDI. So if you don't know the definition of SDI, then you are not going to be able to answer this question. So if you get a question like this, I recommend that you state the definition of Simpson's diversity index, calculate it, and then say, this is the probability of getting these, and just do a statement like that. So make sure that you understand what Simpson's diversity index is. It is probably one of the most crucial parts of a biology data test. The other thing is that you are also likely going to be given the formula for SDI. Definitely memorize it just in case, but it is very likely that it will be given to you. Now, there is one more or 2 more formulas that you might need to know. The first one is percentage frequency. Now, this one definitely isn't too hard, and it's just telling you how often like a species pops up. So you get the number of samples in total. So maybe I took out 10 quadrats. So I've got 10 quadrats that I've taken out, and in 6 of them, this specific plant showed up. So that means if I've got 6 occurrences out of 10 samples, then my percentage frequency is going to be 60%. Now, don't take your 10 quadrats, which are your 10 samples, and then go, oh, in total, there were like 22 of these flowers, and then you get 220% or something. No, that is not how it works for percentage frequency. You're not looking for how many times this one popped up. It's in how many samples did this organism pop up. So you should always get somewhere between 0 and 1, just like any other probability or anything like that. So percentage frequency definitely needs to be between 0% and 100%, and it is the number of times, like the number of samples, and then in how many samples did this species appear. The last one is population density. Now, again, this one might not be very likely, but I'll definitely tell you about it just in case. And population density is simply the population divided by the area, and the area is probably going to be a meter square or something. So that's all that one is. Greater population density means that there are more of that organism in an area. Definitely not as common, but it is something that you might want to consider. Now, another super duper common thing that you will get in your data test, I would definitely say that you're going to get some sort of SDI biodiversity thing. And the other thing that you'll basically certainly get is some sort of classifying ecosystem. And there's really only two that they can actually do. There are a whole bunch of other ways to classify ecosystems out there, but the main one is going to be specs or whatever, however you say it down here. And then, I've forgotten what this one's called. I don't remember what this triangle one is called, but you definitely should recognize this triangle diagram here. I don't know why, but in my head I'm saying like holds or something. Hold on, let me see what classifying ecosystems triangle thing. Yeah, this is what it is. What is it called? Holdridge. I was right, it's hold. So this one is the Holdridge life zones. Now you don't need to know the name of it or anything like that. You just need to be able to classify an ecosystem based on given data and the diagram. Now there is in no way a need to memorize what any of these diagrams are or what each of the classifications are. They will always give it to you. They cannot expect you to memorize this stuff here. So what you'll get is, if you get this Holdridge life zones one, you are going to get 3 sets of data. You'll get like a mean annual biotemperature, which is along here, along the right side. You'll get an annual precipitation, which is these blue lines going down that way. And then you'll get a potential evapotranspiration rate per whatever. So then you will get your red lines going that way. And basically what you do is you just track along, and you can draw on the diagram with your pencil to figure out exactly where this belongs. So this is what I would do for this one. So we have, and I'll just get a bright color, we have a mean annual temperature of 2 degrees. So let's go to the temperature thing and go, okay, so 2 degrees is going to be halfway through between 1.5 and 3, or not really halfway, but somewhere in this little section here is going to be our temperature. We then have an annual precipitation of 900 millimeters. So we would follow these blue lines down and see that 900 is probably going to be in here somewhere. And then an evapotranspiration rate of 0.15. So it's going to be in here somewhere, and you can see that there'll be a crossover in this rain tundra section. So if you ever get 3 sets of data like this, so your 3 data points, and they don't converge at a particular point, then you've definitely done something wrong. They should converge at a particular point, or it is not possible. So if we've got our temperature and our rain, but we had like a really high evapotranspiration thing, as you can see, there is no way that that is going to intersect up there, so you've definitely done something wrong. These can be a little tricky at first just because they're a bit odd, but eventually you will get the hang of it. You also don't need to be that accurate, because as you can see with these lines, as long as you're like somewhere in there, you will get the answer, because these hexagons that are the actual classifications are pretty big, so you will be able to just do like an estimation with your lines. I do also recommend actually drawing on the diagram just in case you get it wrong. That might be shown as some sort of like understanding or working out, and if your teacher's really nice, you might end up getting a mark there. So that is the Holdridge life zones. You must be given 3 different variables, and they must be the precipitation, the biotemperature, and the evapotranspiration rate. And then you just say, oh yes, this all comes together to be a rain tundra, and you draw on the diagram and show that there. The other way of classifying ecosystems is the Australian one, and it is through specs or however you spell it. And it's basically all about the height and the percentage coverage of trees in an area. So you need to do 2 things, and I've got a couple of different example questions here. So we might have a look at this one. What you do is you need to look for the tallest tree. So the tallest tree here is going to be just under 30 meters. So it's between 25 to 30 meters. So that means we've got medium trees in here. It's Cool, so the tallest stratum is medium trees between 10 and 30 meters, which means it must be one of these 4 options in this row here. Now, what you do to figure out the rest of it is you find the foliage cover by the tallest stratum, so the tallest trees here. So sort of like the tallest layer of trees, not the tallest, but like the tallest layer. And usually they will give you some sort of grid, which makes it super easy to do the calculation. So here I would say that these 3 trees are definitely our tallest trees here. So let's sort of figure out what our percentage cover by these trees are. So I'm going to go, and I would definitely recommend drawing on this as well and using a ruler, because it shows that even if you get an answer that's slightly different, it might actually prove that you did do the right process. You were just like a couple of millimeters off or something. So I would say that we've got 2 squares here of coverage, and then we've got 2 here with coverage because we've got 1 here, and then we've sort of got half on each side. So I'd say another 2 squares. And then lastly here, I'd probably go with another 2 or like 2.5 squares, because it's not quite the 3, but it is pretty close. And we do have 10 squares in total. So then that would come to about 6.5 over 10, which would come to about 65% canopy cover. I would then go back to our table and go, right, that is between 30 to 70%. So it is going to be called an open forest, and you would write that down. Now I would write down every step of your working. So you'd say, this is a medium tree because it is the tallest one is between 10 to 30 meters. And then you would say, I think it is an open forest because the percentage coverage is between 30 and 70. As you can see, these are really broad ranges. So often your data test will make it so it's definitely within that range there. So it's like 50%. Well, that means even if you get a little bit over or a little bit under, you're still way in the 30 to 70% range. So you will get that one right. So they do make it easy for you in that way. So those are our 2 main classification processes, and you will 100% get one of them in there. It's basically a guarantee. Now I do have like a whole load more stuff in here, so we're just going to keep yapping. So this one over here, we've got some things that are like sort of like a maybe in there. We're definitely going to get diversity stuff, especially with SDI, and we're definitely going to get a classifying ecosystem. So those are like the 2 that I'm like sure that you're going to get. The rest of this is all like a little bit maybe. So you might have like a 3rd data set with like something else in it. So we're going to go through some of these maybe things now. So first of all, we've got a carrying capacity thing here. So the QCAA definition, which is actually something that you do need to memorize for your externals, says that carrying capacity is the size of a population that can be indefinitely supported by the resources and services available of that ecosystem. And there might be a question on there that just asks you to identify the carrying capacity of an ecosystem, and your carrying capacity is where it plateaus. So that's just where it's rising, or dropping, and it just hits a line. And that's when you know that that's the carrying capacity, because that is just generally what the ecosystem can stand. So what I would do here is, if we're finding the carrying capacity of species A, you grab your ruler and you draw a dashed line across, and you see that it comes sort of at the 7 range. You check the axes for the units, because you don't just say, oh yes, the carrying capacity is 7. That doesn't seem right. 7 is not very high. So it turns out it is number of larvae by 1,000. So we actually have a carrying capacity of about 700 larvae. 7,000 larvae, pardon me. And what I would do is I'd always stick the approximate sign or write approximately in front. The other thing that they might ask you about is like K strategists and R strategists. So that is like the bust versus boom and like all that, like the bust and boom, and then like the more reasonable stuff. So how I remember this is I remember R strategy. So R strategy is like the rats. So if you know anything about rats, they just breed like crazy. So they just breed like millions and millions of little rats, and then they all die off. So that is what an R strategist basically is, like up and down, up and down. They breed heaps, they die heaps, they breed heaps, etc., etc. There's no stability with an R strategy population. And you'll find that the R strategy populations are often very small. They often have massive families. The parents don't usually take care of their young, so it's really just like a fend for yourself, just like a horde of rats. Now, the K strategist, I say K for carrying capacity, because K strategists are those that have a carrying capacity and they get a stable population. And those are for things where there's often like bigger organisms. These organisms might have 2 or 3 children compared to rats would have like 600 children. So they're bigger populations. Like humans are a case strategist. We don't have as many humans. We take a lot of time to take care of our young. We have much longer lifespans, whereas our strategists have very short lifespans, that sort of thing. So understand what a case strategist and an R strategist is, because even though you won't have to talk about the theory and the concepts behind them and what they are, you might need to look at a population graph like this and say which one is more likely to be the case strategist. And I would say species A, because they have a carrying capacity. They actually plateau, like they boom up and then they reach a carrying capacity and they just steadily move along like that. Compared to this one here, which is a lot slower. It's not really like going up and down, nothing like that. So make sure you understand what a K strategist is and what an R strategist is. An R strategist will have a J curve before it drops, and then a K strategist will have an S curve. So definitely something worth looking into. There is another thing here that I did see in some of the practice data tests on the QC Discord server, so nothing official QCAA, but I don't think this is very likely to be on your data test, but it is something that you should know for your IA2s, and that is all about R square value. So we did have these two data things here about light flux and then flora coverage, and we had a trend line which gave us an R square value for both of them. It then says, using the R square value in figure 1, identify the strength of the relationship between light flux and flora. So here it's 0.63, so it's probably like a moderate relationship in terms of strength. Whereas it says contrast the R squared value and the R, of these two parks. So you'd say, well, Tambourine has a higher R squared value, so it's a stronger relationship than this. As I said, I don't think you'll run into this on the data test, so it's not too much stress right now. I don't think you need to worry about it. Just make sure you understand what R squared values represent and sort of like a general, oh, this is a high R squared value, this is a moderate, this is low strength, like that sort of thing. It will be relevant for all sciences in your IA2, so definitely learn about it then, but for the data test, don't worry too much, especially in biology. It's very unlikely that you'll get some sort of thing like this. Another thing that you could get, because it is in the syllabus and it really just depends on like where your teacher is like at teaching you guys, and this is these energy consumption loss diagram sort of things. So there are these diagrams and they are in late unit 3. I don't think it will pop up in your data test just because it is like so late in unit 3 and you probably haven't learned it in class yet, but it is simple to understand, so you might as well, just in case it's on the data test. You typically will have to calculate some sort of unknown. So in this case, the unknown, it doesn't really say it very clearly, is the respiration of primary consumers. So all of these numbers here is the energy that is being used or lost or anything like that, and also the energy that is being passed on. So in this case, if we were to calculate the respiration of primary consumers, you would have a look at this. So we start off with 630 based on that. We then say, okay, we pass 125 on to higher level consumers, so we'll take that away. We pass on 20 to decomposers, and then we pass on 295 to unused. And then we pass off a certain amount, so X in respiration, and all of that leaves us at 0. So we've taken into account all of the energy uses. So they've passed on 125, they've decomposed this, they've spent this or not used this, which means the last thing we haven't figured out is respiration. So basically what you would do here is you would get your calculator, not to your phone, and you do 630 minus 125 minus 20 minus 295, which would get you negative X equals 190. So then you just know that X would be 190. So they've used 190 kilojoules in their respiration. So calculating an unknown is definitely something you could do. The other more common thing is calculating the percentage efficiency of a certain step. So for example, what is the percentage efficiency of producers to primary consumers? So producers start off with 4,400 and 60 kilojoules or whatever of energy. So that's what they start off with. So they got all this energy, but primary consumers are only given 630 of that. So that means the rest of it has all been lost to things like respiration, decomposition, and simply not being used. So to find out the efficiency, you need to find out what percentage of the primary producers have been passed on to the primary consumers. So you would do 630 divided by 4,460, which comes down to 14.13%. Now, most of the time, the percentage efficiency is only in that like 10% range. So if you get a number that low, that is completely understandable. If you get something massive like 80%, something's gone wrong. So percentage efficiency is definitely something that they could ask you, and it should be around like the 10 to 20 range, or just anywhere from 0 to 20 really. Life is very inefficient. Now, what else might we have to do? Okay, here we could be asked about things like density independent and density dependent factors that could limit the size of a population. So like a density dependent factor would be disease, because disease is more ravaging when there's a large population, whereas when there's a small population, it can't really spread as much. You could be asked about abiotic and biotic factors. So abiotic is non-living factors such as climate, temperature, and biotic factors are living ones, so things like disease, predators, etc. You could be given some ecosystem graphs or diagrams, and you'll be asked if these two species are in the same ecological niche. So if you see something like this, where these two species, when they are growing separately, they follow a similar growth curve, but when they're growing together, one of them just dies out. That means that they do occupy the same ecological niche, or majority of the same ecological niche, if they can't survive together. So you might be asked a question like that. You could be asked about the competitive exclusion principle. So that is basically like when you've got two species occupying the same niche, only one of them is going to come out on top. So in this case, P. aurelia is competitively excluding P. caudatum when they are growing in a mixed culture, and it is excluding it, it's killing it out, it's taking over. So competitive exclusion principle is also something you might have to identify from graphs. You might have to say what kind of species relationship a species have based on a population graph. So with this gazelle and lion graph here, when we have a big spike of gazelle, we notice that the lions also spikes and gazelle goes down. This is because the population spike in gazelle gives the lions much more food to eat. So the lion eats it all, grows in population, but then they've eaten all the gazelle. So that means the lion population starts to drop because there's no longer enough food. And then because the lion population is dropping, that means the gazelles can build up again to a big population, which then starts the cycle all over again of the lions and eating it all, growing in population, gazelles dropping, etc., etc. That is an example of predation. So whenever you see something that looks like this, with massive spikes followed by little spikes of another bigger species, that is going to be predation. So you might need to figure out the relationship based on a population graph like that. And the hint is it's basically always going to be predation. Other things you might do, you might also be asked to describe what would happen to a population or a graph if something is changed. For example, if we add more food to the system, maybe P. aurelia will spike up even more, or maybe P. caudatum could also continue to grow because P. aurelia is no longer eating all the food. Or maybe if we introduce a vaccine for a disease, then population might go up. Or maybe if a disease is introduced, then population can plummet. Another thing might be keystone species stuff, but honestly, there's not a lot of data involved with that, so I doubt you'll get asked very much to do with that. Okie dokie, not much longer to go. I know this has been a super long video, but that is just the nature of biology. Biology is all about yapping. If you do want me to do in-depth question answers for all of these sorts of things, let me know in the comments. Just ask me. I'm more than happy to see if I can give it a go. Okay, so p-values. P-values are pretty important, and they're important for your IA too as well, and they're just about determining about whether or not there is a significant difference between populations in two different environments or habitats. So say we go, okay, there's like 50 lions here and 20 lions here. Do some t-test p calculations and you get given a p-value. to see if there is a significant difference. Now, in your IA2, you'll have to do like the t-test and stuff yourself to figure that out, but not in the data test. There's no calculating involved. You'll just be given a p-value. Typically, you need a p-value of under 0.05 for there to be a statistically significant difference. Now, the boundary for that is determined by the researchers or whatever. Now, that is not something you need to worry about in year 12. It's like a university level thing. So just go for always below 0.05 as the threshold. If it is less than 0.05, then there is a legitimate difference between population numbers. Now, I do have some things up here that I think you should be studying just as a bit of a checklist, and there might be more that I have missed out, but species abundance, species evenness, the Lincoln Index, Simpson's diversity index, and what it actually means, percentage frequency, classifying ecosystems based on data, abiotic, density dependent and density independent factors, how and why do they affect a population, and how do you decide what is a relevant factor based on the data. So in a jungle environment, is water temperature going to be relevant? No, that's probably more relevant for an ocean environment, whereas a jungle environment is probably going to be more relevant to sunlight or canopy cover. Relationships between animals, and more importantly, can you determine what relationship it is based on a graph? So relationships include predation, competition, mutualism, commensalism, amensalism, parasitism. All of these are stated in your syllabus, so have a look at that. And the competitive exclusion principle. So are the organisms out competing and pushing one of them out? And you can see that in things like this, of when they're growing separately versus when they're growing together. Now, what are some common question types? Contrasting things like contrast species richness, species evenness, SDI, all of those things between areas. Calculating species diversity, Lincoln Index, determining the probability of finding the same species, and justifying your response. So for any data test, whenever they say justify, state actual values from the table or graph, etc., etc. Now, I do have some final tips here. In every single response, make an effort to include evidence from the data. This is a data test. Everything should be backed up by the data. The formulas that you're most likely to see are going to be given to you because you don't have a formula sheet. I do suggest memorizing them anyway, just in case, but they very likely will be given to you. Be realistic with your practice. If you haven't done it in class yet and you ask your teacher, will we learn this before the data test? And they say no, it's not going to be on the data test. Don't be afraid to ask your teacher, like, do I need to know this? They'll very likely say, no, you don't need to know this. Oh, maybe you should study that. Use your teachers, talk to them, ask them about what's going on. Cognitive verbs are very important. So do you know what it means to identify something or contrast something or compare something? If you don't, I do have a whole video on that, which I'll link in the description. You can't answer a question if you don't know what it is actually asking you to do. For example, compare. Compare means state the similarities and differences. Most people just state the differences, but you also need to state the similarities or you won't get the mark. Make sure you know what the question is asking you, and you do that by knowing what the cognitive verbs mean. Relationships in biology aren't going to be mathematical. Write a word response for relationships when they ask you. And lastly, I can make extra videos if you want to see me actually do practice questions or if you think I've missed anything out. Thank you so much for watching, and hopefully this helps you get a 10 out of 10 on that data test. I definitely have faith in you. Contact me through the comments, through my email, or through Discord. Any of that works. I hope you like this video, and stay tuned for chemistry next week. Just ask if you have any more questions, and to support me, please like, comment, and subscribe just so I can keep making videos like this for you. But yeah, thank you for watching.